All right, so now we're going to move on to Chapter 7 and talk about the other branch of the immune system referred to as adaptive immunity. And so remember from Chapter 6, the first branch we talked about was the innate immune system. So we're going to talk about there are a few similarities, but there are a lot of striking differences really between the two immune systems. So adaptive immunity, its purpose, again, and so this is somewhat similar to the innate, but it's destructive of infectious microorganisms that are resistant to inflammation, right? So it's when the innate immune system basically does its thing and can't do enough, all right? Then the adaptive immune system kicks in. So it's typically second in terms of time. The adaptive immune system is long-term, right, whereas opposed to typically the innate is unless there's a disease, typically the innate is relatively short-term or short-lived. Highly effective protection against future exposure to the same microorganism, all right? And so what that is, that the keyword there is the same microorganism, right? Meaning that it's very specific, all right? There are different types of different parts of the innate immune system or different cells in, adaptive immune system are targeted specifically to a specific type of pathogen, okay? Which makes it very specific, right? Long-lived, you already said. And therefore, because it's future exposure, right, that's another keyword here, we have something called memory. Okay, so if it's like vaccines, for instance, take example of that. You get a vaccine of perhaps a inactivated virus, right? That can't harm you, all right? And then what happens is your body ramps up an immune system or an immune response, and remembers that, right? It doesn't ramp up an immune response, but it prepares itself for the next time that it actually encounters that. And typically, what's going to happen is when you're actually going to encounter the actual real pathogen, and your body's already ready to fight it off relatively quickly. So there's two types of the adaptive immune system: active immunity, which is the more common one, right? So again, the antigen, we're always going to be needed to exposed by some antigen, which is a part of a pathogen. It could be a bacteria, a virus, fungus, anything. Something that the adaptive immune system recognizes as not part of the self, right? So we have self antigens and non-self antigens, and we'll get into that. And again, this is what we just talked about, the immunization. So, or it doesn't have to be immunization. It could be you get sick with something like chicken pox or something, and then you have a memory to that disease in the future. So you didn't get necessarily an immunization to it, but you were exposed to it naturally, and now you have an immune system devoted specifically to the virus that causes, say, the chicken pox. And then passive adaptive immunity is when your body, so with active immunity, your body makes its own immune response. Makes its own immune response. Passive means that you're getting preformed antibodies or T cells. We'll talk about those. Those are really the two components of the adaptive immune system being administered exogenously through maybe an injection or something like that. So we're making these antibodies or these T cells somewhere else outside of the body and then using them as the treatment. So again, coming back to antigens, we'll talk about these in a minute, again, in a diagram, but these are important for kind of allowing the immune system to recognize something as not being foreign. They bind to antibodies, and they also bind to T and B cells, which are the main type of cells. They're not necessarily immunogens, but they can be, right? So immunogens are things that create an immune response. So self-antigens would be an example of something that's an antigen, but not an immunogen. So the immunogens are something that maybe would be on a bacteria or a virus, something that doesn't belong, or even if you got the wrong blood transfusion, right? You're adding human cells, but they're from somebody else, and they don't. Your body recognizes them as foreign. So all immunogen means that it's recognizing as foreign, and it's going to be creating an immune response. These immunogens again create the production of these parts of the adaptive immune system, and again, kind of what I just said, but all immunogens are antigens, but not all antigens are immunogens. And haptens are kind of a small subset of antigens, but immunogens are too small on their own, or not sufficient enough to be an immunogen. They can't generate their own immune response. What they do is they combine with a carrier molecule. A lot of times it's already in the body, and together, the two parts come together and they function as a full immunogen. So the poison ivy, what causes the rash in poison ivy itself isn't big enough, but when it combines with a protein within the skin, two together are able to bind to and activate T cells. We'll talk about that later on, and act as an immunogen. All right, so again, antigens are important, but they have to be often presented to the immune cells that are part of the adaptive immune system. All right, so we have these things called MHC, or major histocompatibility complexes. They are glycoproteins on the surface of all human cells, but other cells as well, except for red blood cells. Right, so these are things that are sticking out on the plasma membrane. All right, they're also referred to as HLAs, and they can be broken up into two categories. Right, so class 1 is over here on the left. These are present on all endogenous, meaning from your own body, on all endogenous antigens. All right. So these are expressed on all nucleated cells and platelets, expressed on all, and what they do is these cells present endogenous antigen to tell a cell that, a T cell, specifically a CD8 T cell, that a cell is infected. So if a virus is infecting your own cell, that virus is infecting the inside of the cell. And a T cell may not necessarily recognize that host cell as being bad because it can't see inside the cell, essentially. So what these things do is they they become expressed as on a cell that only when after it's been infected to tell CD8, these T cells, that it's infected and we need to get rid of that. All right. Class 2, what they do is they present exogenous antigens from cells outside the body. So these are only expressed in macrophages, dendritic cells, and B cells, right? And they're recruiting the other kind of T cell. We'll get into that in a minute. But these are important because remember, antigens are what are going to be creating an immune response, right? It's going to allow us to recognize what's normal and what's not normal, right? And allow a response to be generated. Okay. So again, we're going to continue to break down the adaptive immunity. Remember, we're still adaptive, and we're going to break it up really into two kind of spots, although they're related to each other. Again, antigens are the important thing that are on cells or other or outside things that allow for recognition and a lot of times the activation of the adaptive immune system. Right? Lymphocytes, right, which are a type of white blood cell, are the important cells of these, or the kind of the more important anyway. Then we can further break them up into different groups. The two major groups of cells are T cells and B cells. These are both types of lymphocytes. Okay, and then there's other components. So kind of a lot of times we can further break up this immune system into different ways. Humoral, which are really the antibodies. These are things that are made specifically by a modified type of B cell. All right, and so what they're doing is helping the immune system by binding to antigens on specifically bacteria and viruses. So these are proteins, and we'll talk about them later on. And then there's a cellular component. So the B cells themselves that really don't do anything. They don't do anything. But specifically, they're part of the humoral component. They make antibodies or involved in antibody making. T cells themselves are their own component, a cellular component. There are two subpopulations. We'll talk about them. One directly kills cells. The other one stimulates other white blood cells. And then both T and B lymphocytes make memory cells, right, to remember that infection for the future. Right. So again, if we look here, if we focus on the left side, which is going to be the T cell side, all the basically precursors to both T and B cells, so in this case T cells, are made in bone marrow. T cells are not fully mature until they travel from the blood in the blood after the bone marrow to the thymus, where they become mature. They then migrate T cells leave the thymus and go to lymph nodes, where they kind of wait for an infection. All right, and then they can differentiate into these kind of different types. We'll talk about a few of them later on. All right, some of these T cells actually directly kill their effectors directly, or their targets directly. Others help to activate other parts of the system. All right, and so one of the things that they do is they can actually activate the other side of the pathway. We'll talk about which ones do which in a minute. B cell side, right? Again, bone marrow. B cells are already mature in the bone marrow. They then go to lymph nodes, and then when they're we'll come into this in a little bit more detail later on in the presentation. But eventually, when they're exposed to an antigen, whether it be presented or something like that, they become plasma cells which produce antibodies. All right. Some of them become antibodies, some of them become memory B cells. These antibodies are important for destruction, and then the B cells become memory cells. So if we start out with just the T, right, T cells, remember T is involved with thymus. These ones, specifically we're going to talk about Cd4 positive, all right? So these they're helper T cells. They help antigen-driven maturation of both B and T cells, right? So if we come back here, right? This one right here specifically is CD4 positive T cells, one specific type. They facilitate and magnify the interaction between antigen-presenting cells and immunocompetent lymphocytes, all right? And then even within CD4 positive cells, we call them CD4 positive because, well, we'll kind of get on the next slide, but they express a protein called cluster differentiation 4, so CD4, on their plasma membrane, right? The other types of T cells do not express this. So Th1 CD4 positive cells provide help in developing cell-mediated immunity. And what we're going to talk about later on is Th2 CD4 positive cells help to basically develop humoral immunity through B cells and plasma cells. Okay. CD8 positive, right? So again, here this green is the CD8 positive cell. Right, so it's a different type of T cell that expresses a cluster differentiation number 8 protein on its membrane, and it's just this pink one here, sorry. And what it does is it recognizes an MHC class 1 on an infected target cell, all right? And so when it does that, and each of these T cells recognizes one type of antigen, right? There's one for an antigen maybe in a streptococcus bacteria, there's one for every type of virus has a different one, right? Each one has its own specific type of CD8 cell and responds to a different class 1 MHC cell. So what they do in response to that, when they come in contact with an MHC cell that they recognize, class 1, or MHC molecule they recognize, they release perforin granzymes and other types of things, but really these two enzymes, or these two molecules, which basically do what they say. They perforate the host cell. They cause holes to form in the infected target cell. And what that does is basically causes its cell to die. And so that's why they're called cytotoxic T cells, because they actually kill cells, or TC cells. Clonal selection. So clonal selection is basically when a B cell gets activated, it makes multiple clones of itself. All right, and so we'll talk a little bit about that, right? And again, the key thing is an antigen. So every B cell recognizes a different antigen. So an immunocompetent B cell encounters an antigen for the first time, right? For the first time. If it happens in the future, it's a slightly different process. What happens is the B cells, so they have to have a specific B cell receptor. What you'll see is that each of those B cell receptors is an antibody. Right, so B cell receptors are just antibodies that are embedded in a B cell. Once they're released, they're called antibodies. So every B cell expresses the same antibody, which only binds to or recognizes one antigen. And so when the B cell with that specific B cell receptor binds to an antigen, it becomes stimulated to essentially differentiate and proliferate. All right, but before it can do that, it kind of needs we don't want to generate a faulty immune response. We want to make sure that we're generating an immune response. So we almost need a checkpoint. So these Th2 helpers, remember these are the CD4 positive Th2 subtype, right? Basically, these T cells essentially give the, okay, let's go ahead to the B cell to start to produce an immune response. And so what happens is that through the after the antigen binds to the B cell receptor, then it expresses the part of the antigen that would be here through MHC class 2. So remember, these are ones that are expressed only on a few type of cells, including B cells. And it expresses it to the CD4 T cell through its T cell receptor. And if the T cell says, yep, that's good, let's go ahead, let's generate an immune response, the T cell releases interleukin 4, which then goes back to the B cell and says, all right, we're ready to go. That's the symbol. And once it does that, these cells differentiate into what we call plasma cells, and they usually multiply. They multiply into multiple, so they do increasing number of plasma cells. And the plasma cells then are B cells that then release the receptor. All right, they release the B cell receptor to become antibodies. All right, we'll talk about what these antibodies do later on. So again, over here, a differentiated B cell becomes a plasma cell. Then a plasma cell is basically a factory for antibody production. It just generates crazy amounts of antibodies and spits them out of the cell into usually the blood, where then they can go and do other things. The other thing that they do, which is not mentioned here, is they become, some of them, a few of them become memory B cells. So the next time if you ever encounter that same antigen, you're able to generate a response a little bit quicker. But the plasma cells are kind of the key thing. They're the differentiated B cells that generate and produce antibodies. So what are antibodies? Well, again, antibodies kind of have this kind of Y-shaped structure, and we'll come back to them in a minute. We can further break them up into 5 categories. IG something, right? So IgG, IgA, IgM, IgE, IgD. IG gamed, right? If you kind of put them all together, GAMED. They have some similarities, but also some differences based on their structure and their function. So in general, right, we have a couple different chains. Our generic antibody. We have 4 polypeptide chains, right? 2 light ones and 2 heavy ones. So these ones that say the green that say Cl, each of those is a light chain. CH is a heavy chain. All right. We have our antigen binding fragment, or FAB, all right, which is this kind of region all up in here, all right, which is made up of these FRs, which are server recognition sites. And so these things, this part at this end, the FAB, is what's different with every antibody, right? So not every antibody, but even within the same class, it's this region which allows the antibody to bind to specific antigens. Okay, so this one could only bind to an antigen that has this specific epitope, right? If it had any other look to it, an epitope is basically a shape of the antigen. So this antibody can only bind to one antigen. If it's a different antigen, it needs a different antibody. Okay, so but where the antigen and the antibody interact, whether it be as a B cell receptor on the B cell, or it be after the plasma cell has released those antibodies, those B cell receptors as antibodies into the blood, the FAB stays the same. All right, so but again, those basically determine what antigen it can bind to, and we call it part of the variable because it varies between antibodies. And we also have the constant region, the FC region, which determines the biological function. We'll get back to that in a minute. But all the antibodies within a given class have the same FC region. So every IgG molecule has the same FC, right? Every IgE molecule has the same FC. It's different than the IgG, but they all have the same. And if you look within, say, all the IgG molecules or antibodies that are out there, yeah, they all have the same FC, but what they have is different variable regions, right? Different antigen binding fragments. And again, with the antigen and antibody interaction between the antigen and the FAB region, it's kind of like a key into a lock, right? There's for every lock, there's only one key that opens it up, right? One shape of a key. There may be many keys, maybe we might have copies of that key that you go at the store and get that made, right? But each of those copies is identical, right? But if you have a different key with a different style, right, it won't open up that door. And then there's non-covalent, really disulfide bonds that hold these together, and they're important for maintaining the structure of an antibody. So we'll go through some of the categories now. IgG, or immunoglobulin G, this is the most abundant. It accounts for most of the protective activity against infections, and this is one that's actually transported from the mother to the father, or sorry, to the fetus. It's expressed as a monomer. You just see one at a time. We'll talk about some that are not. You see groups them together, but IgGs, this is one whole molecule. They tend to be by themselves, not bound to other IgG molecules. Remember, FC here, constant region, FAB over here, right? So through the variable region, that FAB is binding to an antigen. Okay, IgA, there's 2 classes, but these are found predominantly in the blood. Sorry, IgA2 are molecules that are in body secretions. These are actually the most important. So what we see is that IgA actually occurs as a dimer, meaning that there's an example of an even bigger piece with IgM, but it occurs as a dimer, meaning 2, right? So you see here's one molecule, here's 2, right? They're identical, and they're attached to each other through a J chain and a secretory piece. All right, what the secretory piece likely does is functions to protect the IgA molecules from enzyme degradation, to keep their half-life increased. IgM molecules, these are the largest, all right, and that's because and what they do is they serve as a pentamer, also stabilized by a J chain, slightly different, but pentamer meaning 5 identical IgM molecules bond to each other. This is the first antibody produced during a primary response to an antigen, all right, and they tend to be synthesized very early on in neonatal life. So the fetus actually makes them. IgD, these are kind of a low concentration, and ignore this, but they identify when an immature B lymphocyte is ready for activation when they're expressed. And these 2, like IgG, are expressed as monomers. And then last, IgE, these are the least concentrated. They are involved or mediate allergic responses, and they're also a defender against parasites. So they're associated with, eosinophils for here. And they're associated with basophils, often up here. So what do these antibodies do, right, once they're released? Well, they do, depending on what they are, a number of different things, either direct or indirect. So for instance, with a virus, and some antibodies are involved with neutralizing the virus. So here you see a virus binding to and infecting a human cell. The antibody basically blocks that virus, so physically gets in the way and doesn't allow it to bind to the receptor on the target cell. So it can also do that with bacteria. It can be it can neutralize the bacterial, not necessarily the bacteria itself, but maybe the toxin that's producing to infect the cell. It can, through complement, do complement-mediated killing. So remember complement from last time, so it can actually start to activate the innate response to activate that complement system. And it can also be helpful for phagocytic cells. It kind of recruits phagocytic cells to their, say, bacteria, right? When I've heard this described as it's kind of like a salt, right? It makes food taste better. So we put salt on, we sprinkle the antibody, which is the salt, onto a bacteria, and then the bacteria, the macrophage is more tasty than the macrophage, and it wants to eat it up faster. And then, so in response to a kind of a bigger picture thing here, in response to some kind of an immune response that generates the adaptive immune system, specifically antibodies, we have the primary and the secondary. So early on, obviously the primary, that's the initial response. There's a latent period or a lag period. So here, when you get exposed to the antigen, and then, so on the y-axis, we have the essentially antibody titer. Maybe you've heard that in the news. That's the amount of antibody you have at any given time. So right now, MMR is a big one. People that got MMR vaccines that generated maybe had the MMR, different antibodies to those viruses. They're supposed to last the rest of their life. At least that was the plan. But now they're realizing that their antibody titer is low. They don't have enough. They don't have the antibodies that they've lost. They've lost antibodies over time. They have to get a second shot. But anyway, so this describes the amount of antibody titer, right? And then in the x-axis over time. So the primary response, right? So first we have the first one we have, at least the first peak anyways, IgM, which we mentioned before, followed by a slower IgG, which is more sustained, right? In the future, right, secondary response would be if you got the vaccine, right, for something, and then eventually you got actually exposed to that virus. So the second time you were exposed to that, you have both IgM and IgA start to rise at the same time, but you have a massive, very, very robust IgG response that you in a relatively, although not, but IgM, relatively small IgM response. And then some other cells that are involved with the adaptive immune system, NK cells, at least that are have a role. They may be, actually, a lot of them are part of the NK cells are actually also part of the innate system, but they complement the cytotoxic T cell mechanisms, lymphokine secreting T cells that amplify inflammation, and T regulatory lymphocytes, or we call Tregs, which provide peripheral tolerance and suppress the immune response. Pediatric immunity, so in children or the fetus has sufficient IgM, but it's deficient in IgG and IgA in particular, so we have to get them from the mother. So it makes its own IgM, but these two here we need from the mother. And immunologically immature fetuses or babies when they're born with deficiencies in antibody production, phagocytic activity, and complement activity until later on. This is one of the reasons why you have to be careful with giving vaccines to a fetus that's too young. And then on the opposite end, there's obviously aging and immune function. As you get old, at least from fetus, as you start to get older, your immune system gets better. But as the person ages, the immune system tends to decrease in activity. So we have, as a person ages, decreased T cell activity. Their thymus gets smaller, 85% reduction in size, so that's less able to mature T cells. Thymic hormone production drops, as does the organ's ability to mediate differentiation. And then there's also a decreased antibody response to antigens.